Good morning, everybody. Yeah. All right, just looking through and doing the usual with attendance real quick. Anybody have any questions so far now that we've got like kind of cranking a little bit with some of the online work? Kristen, Erica. Jackie and Emma. That's it for now. It looks like we only had 2 people that didn't get on to Top Hat and complete their pre-lab, and they are 2 people that haven't even shown up yet for class. So I'm not too concerned about that. I hope that it was easy enough to kind of figure out once you had finally got into Top Hat. That must have been a treat. So but now that it is working, we have an idea about it. It should be a lot easier to go from there. I don't have any way to look at Top Hat the way that you guys do, so I can't ever see it like how you guys see it. And I just don't have an interface like that. And I can't even get on it on my phone at all. So there might be times that you guys could be having an issue with something on Top Hat, or you want me to like open up an assignment on Top Hat, which is, completely different from opening it in Canvas, because I have to do it through Top Hat. I can't do it on my phone. And so that will limit me whenever I'm off hours. And in the summertime, I'm mostly off hours, right? So if I'm not in here like lecturing to you guys, or like it's not right after, class or something, typically I'm off hours. So if you do have any issues like that, just be patient, please. And I also you'll get grades in Top Hat, and you'll notice they aren't going to be present in Canvas. It doesn't automatically transfer over. It's not like Connect with McGraw-Hill. So if you guys have done Connect before, which I know a lot of people have, it's not like that. It doesn't automatically transfer. I have to manually type your grades back over into it. They just don't have the interface. So that's why I don't transfer the grades, especially with you guys doing, labs pretty much every day. Your grades aren't going to be transferred immediately. I usually do it once a week. Usually at the end of the week, I'll do the whole week all at once. So just be aware of that. And I know there's notes about it in Canvas, but I don't want anybody to get confused. I've got Marissa. We got Ashoria. Don't have Destiny. Doesn't look like it. Okay. Cool. Well, so today would be our first day of actual lab work if we were able to be in the lab. They do have the lab open now. Like I'll say, like we can go, you really so much, and I don't really even want to go in there, but people can go in there now. They don't have the big old shower set up anymore. Like we couldn't even get on the floor at all. So now at least the doors, you can access it, but they're doing so much construction and tearing things down. It's not safe on the second floor. They're building a whole new lab in there. And like I said, it's a biotechnology, biomanufacturing lab. So it's a new program we offer and it's great, but in biomanufacturing, you have like clean rooms and you have specific stations for this and that. And so they're like installing a whole bunch of stuff that is making it really difficult to get to the microbiology lab, and which is immediately next door to it. And they didn't really give us a heads up on any of that. So that's why we aren't prepared. It's not really our fault. They sent us a schedule about a week before they were supposed to start the work, and then they started the work like 2 days after they sent the schedule. Was not at all like so not at all whenever they were supposed to, and they didn't start where they were supposed to, so we didn't have any time to move anything. So I apologize for that. It was completely unforeseen. We didn't know they were doing any of this. We knew they were going to build a new lab, but we didn't know they were going to tear down the rest of the building for asbestos. So I mean, they say they're doing asbestos cleaning, but they haven't done it in the offices. Are they just going to leave it in here? I don't know what's going on anymore. I don't know if they're going to like close down the offices later. I know they're going to tear out. It doesn't matter. It's not your problem. So, but yeah, it's a good time is what I'm saying. So yeah. I don't know what that noise was. Sorry, if you guys are talking, I had my volume turned really far down. So if you guys said something to me, I don't know if you did, but it wouldn't have been your fault. It would have been mine. Let me double check before we go. Okay. So whatever. So this was gonna be our first lab, first time that we were gonna, actually handle bacterial growth media, which if you are actually following along on the lecture schedule, then you've probably been introduced via, chapter 3 into the tools that we'll be using in microbiology, things like media. We mentioned it a little bit in the last talk. Basically just stuff that bacteria will grow in or on in a controlled way, right? Provides them the nutrients they need and in a fashion that we can use them if we need them in a liquid form or if we need them on a solid surface for whatever reasons. But anyways, so we were going to get introduced to that and get to actually use those hands-on for the first time today in the lab, where we would have actually gone in person through like lab safety stuff and all of that. So great. So we didn't get to do that, but we're going to go ahead and start with this material anyways. These labs that we're talking about and doing, I'd say doing virtually, aseptic technique and isolation, that's easy enough to talk about and see pictures of and understand it. When we get into the stains, like the gram stain or the acid-fast stain or anything like that, those, you guys, it's not as simple as just like knowing that the gram stain can tell the difference between gram-positive and gram-negative bacteria. It's not that. So you'll have to know what each step is. So I don't care about how long you put the stain on. I don't care at all about that. But you do need to know that the first stain that we use, the primary stain, what would you think primary would be first, right? Number 1. And that's true. That's exactly what it is. So the first stain, the primary stain, is crystal violet for that one. And you would need to know that, and you need to know why we use crystal violet. What is the purpose of the crystal violet? How is it working and serving its purpose in that stain? So that's the sort of stuff you need to be learning from the labs. And it's just so much easier to understand what you're doing when it's hands-on. So even though we might have to have those labs up front virtually, when we get the lab back and working, some of those we will have to go back and redo by hand so that you can get the experience with them. And it might seem to you like, who cares about this and nobody's going to use it? But if you're going into nursing, one of the most important things you can understand about treating your patients who have infectious diseases is that if somebody is interested in whether it's bacteria or virus or fungus or whatever, one of the first things they're going to do with whatever sample you collected is run a gram stain. So they're going to want to look at it with a stain on it to see, is it gram positive, is it gram negative, or is it not a bacteria, and gram stain's a real easy, quick way to do that. You have a patient that comes into your ER and is coughing up blood. It's a homeless man, and he's coughing up blood and sort of greenish sputum. That's the stuff that you cough up, and so you would collect some of that sputum, isn't that wonderful, in a little cup, and then send that to the lab and probably order an acid-fast stain. The acid-fast stain looks for acid-fast, go figure, bacilli, which are rod-shaped bacteria. And acid-fast bacteria that are rod-shaped, that are found in sputum, if you tested positive for acid-fast bacteria, would be Mycobacterium. Typically, Mycobacterium tuberculosis, like we mentioned, that causes tuberculosis, but it could be Mycobacterium avium. If you have HIV, you can have tuberculosis present with Mycobacterium avium, and go figure that avium is found in birds, right? But when you're immune compromised from HIV, it can cause tuberculosis, but with a different bacteria. So stuff like that tells you a lot. And it is super quick to do those tests. And a lot of people are like, well, just run the PCR. PCR is not super quick, and PCR does not work the way that you think it does. I promise you. So a lot of people think you just swab somebody and pop it in a machine and it tells you an answer, and it's not that simple. But we'll get into that when we get into the DNA stuff. When we talk about DNA, and you're going to need to understand DNA and its structure in a way that you've probably never learned about it before, unfortunately. But when we do, then you'll get why PCR isn't a magic fix for everything. You have to know what you're looking for. So you have to do PCR for COVID. And if you put your little swab into the thing and do the test for the PCR test for COVID, it's not going to tell you if you have flu or Ebola or whatever else. It only will tell you if you have COVID or not. Yes or no on the COVID. If somebody comes in and they are bleeding out all over the place and you take a swab of their blood and do a PCR test on it, you think it might be Ebola and you're, I don't know, somehow magical and you have the ability to test for Ebola, it's not going to tell you if it's any other hemorrhagic disease. It's just going to tell you if it's Ebola or not. If you even have the test for it, it's not like you can just dial up any test you want. So we'll get into that when we get into DNA, but I think it's very important to understand that. And plus, PCR doesn't, it's not super fast. It takes a minimum of an hour to get an accurate result for most PCRs. And even then, in most other industries besides medicine, they're going to say it's more like 3 hours, just because of the sensitivity and everything like that. So PCR is not what you think it is, probably. So these other tests are actually leaned on a lot more than you might think. And you'll probably learn more about that as you guys are going into medicine or working in the medical field. I'm assuming most of you are. If you're not, then cool. But I still have to teach you this stuff. So this is kind of our bread and butter. Unit 1, chapters 1 and 3, the intro chapters, right? The chapter 1, basic idea about concepts about microbiology. Chapter 3 is the tools we use. Chapter 2, reviewing chemistry. And yeah, you really do need to understand chemistry for this course. And it's going to start flying out of my mouth like nobody's business all the time. So if you haven't, and you think that you're fine and you did okay in chemistry, you don't need to brush up on it. You're probably wrong. You probably do need to brush up on it. Because already I was spitting out words like hydrophilic and hydrophobic and amphipathic and all these things, and those are going to come up often. So please take the time to understand your chemistry or review it if you need to. If I say something like peptidoglycan to you, and that doesn't even mean anything, even at face value, then you definitely need to review your chemistry. I'm not saying that specific word, but if you can't even break down that word into its components and that means something to you, you need to review your chemistry. Peptide. Have we talked about peptides in here? No, maybe not, unless you've watched, chapter 2, but you should have in your chemistry courses. Peptide bond is the bond between two amino acids. It's a bond that happens between an amino group and a carboxyl group. Linking up two amino acids together to form proteins. So that's a peptide bond. So peptido, you have peptides there, so probably little chains like mini protein chains, right? And glycan, what would that make you guys think of? It sounds glycan to me sounds a lot like maybe sugary, think of glucose and stuff like that. So glycan is in fact a sugar. You've got repeating sugar units linked together by peptides, peptidoglycan. It's not that hard once you break it down like that. But if those words aren't familiar enough for you to pull it out of there, that's what I mean by you need to review. I need you to draw me pictures. I will never need you to draw me pictures. I'm not going to put up pictures that are so similar to one another, you'll never tell the difference and ask you to tell me the difference. But just be familiar with these terms. Peptidoglycan. Oh yeah, that's the sugars that are in chains linked together by the peptide bridges. Yeah, and that's what makes up bacterial cell walls. And whether you have a thick cell wall or a thin cell wall determines whether you're gram positive or gram negative. Because if you have a thick cell wall, the thick cell wall traps that crystal violet dye that I was just talking about, traps it in there. The thin one doesn't hold it in there. So when you do your rinses and all of these things that we're going to do, the crystal violet, which is purple, go figure, gets held into the gram positive and not in the gram negative. That helps you tell the difference between the two. And why would that be important? Well, a lot of gut-based bacteria are gram negative, and a lot of skin-based bacteria are gram-positive. And a lot of these pathogens are divided between gram-positive and gram-negative, and you treat them differently because of their traits related to that. So it's actually the absolute number one most important defining characteristic between bacteria, whether it's gram-positive or gram-negative. So that sort of stuff, I'm sure you start to imagine. Really important to actually get the hands-on work, but we're going to still crank through these on schedule. And then later, when we get back into the lab, we will add in our gram stains on in person or something like that, or at least at the barest minimum, walking through it together while I'm doing it, and you guys can follow along in that. And we won't do a whole lab about it, but at least get you hands on about it because you maybe hopefully already get the concept. That's what this is for. So hopefully it's a mess, but hopefully that will be good enough. That's what we're going to strive for. It's not ideal, but it is what it is, right? This is for aseptic technique and isolation. You can't do any of these exercises I keep saying about putting the bacteria here and growing it on the media there, and then we incubate it and all of this. And obviously, all of this stuff has to be done in a controlled environment. Aseptic technique. That's the technique that we use to prevent contamination. That's a general way of looking at it. And so prevent contamination. Like if you're, I've already, freaking, here it is. This one's broken, but if you had your sample and you want to keep it just your sample, let's say it's your sample of staph. You're trying to look at this particular strain of staph that you collected from your patient. It seems to be resistant to certain antibiotics, and now you want to investigate what antibiotics you can use. So you don't want it contaminated, right? You need to know about your strain, not other crap from the environment. So now maybe you got it isolated. You don't want the outside stuff getting in, but you also don't want to get this resistant strain of bacteria everywhere else around you. So avoid contamination into your sample, avoid contamination outside of it as well. Yeah, that's a pretty simple concept. So that's all aseptic technique is. It's preventing contamination. It does not always have to deal with pathogens. If you were just a scientist studying some random soil bacteria that doesn't cause disease, you still want to prevent contamination, yeah? So aseptic technique, anytime that you are dealing with any sort of potential risk of contamination and trying to prevent it, yeah? And that does include anytime you're not dealing directly with growing bacteria. So we'll get into that a little bit, but like dealing in a healthcare environment, right? So anyways, so I said that you got your little sample and your patient comes in with a skin wound. Maybe they, cut their arm and they didn't wash it well enough. Or like me, when I was a little kid, I went rollerblading and I fell and I scraped my knee. And I didn't tell my mom because I didn't want to get in trouble because I wasn't supposed to be rollerblading. And but I did it anyways. And then I didn't tell mom, didn't wash my knee because I was a little kid and I didn't understand. And then it got infected. And mom was not happy, but it got infected. So, you get that yellowy, greenish gooiness to it. Sometimes, depending on how bad this can get, you might have smell, you might have redness and hotness emanating kind of from that area and everything like that. We know kind of what it looks like. Take a little swab, a sterile swab, you as a nurse might, swab that sample of the pus or the area that's wounded. And we're going to send that swab to the lab. Now, when you swab that wound, do you think, let's give it this, that it's just one bacteria causing the actual infection, okay? Do you think that when you swab the wound that you're only picking up that bacteria? No, you're picking up all the bacteria from the skin, whatever else went into the wound, everything. So which one is the one causing your infection problem? So you might want to isolate it. So it's going to be a mixture. Or we could go back to what we were saying before with the ubiquity of microbes, swabbing the bottom of your shoe. If you got a whole bunch of different colonies and you wanted to just study one, the orange ones, for example, how would you go about isolating it and separating it out from the others to make a pure culture? A pure culture is a culture that consists of only one species of bacteria. And you know what it is typically, but it's okay if you don't. It can still be pure if you don't, right? If you're trying to figure out what it is. But if you know that it's pure, you came from one colony from the plate, typically almost always that's going to be pure because colonies come from one cell from one bacterial species, one colony. So if you can select out one colony, should be good. Okay, why is this not going? Okay, so getting into it. So we talked about aseptic technique and what it is, preventing our contamination. Cultures, what is that? It's basically just our ability to grow up whatever that sample is that you're trying to investigate. So it means to get more of that sample to do our testing on. Because like you swab somebody's wound and you don't want it to spread so much, or there's so much sample that you could work with it easily, right? You want to get a little bit, whatever little bit there is from that patient before it gets overgrown. And then you can grow it up in the lab and do all your experiments, right? You don't want to just keep going to the patient and getting more. So you work with your own culture. Sterile is a term that means devoid of any of microbes. Oh, I have the answers on the next page. Let's just pull it up. Here we go. All right, so sterile means that it is devoid of any and all microorganisms altogether. So there shouldn't be any living cellular material on there. As we go through all of our chapters and you get introduced to viruses and prions, this gets a little bit tricky because no, those are not alive, literally not alive. So how do you determine if it's sterile if you can't even tell if it's alive or not, right? So usually with viruses, we would say that they are inactivated. They can no longer be, active and forming infections and stuff like that. They're not alive, but they can't live outside of your cells at all. 0% living outside of your cells. They can't do anything outside of your cells. They have to be in your cells using your cellular machinery in order to grow. That's what viruses do. But it's more complicated than that, of course. And then prions are like next level. So if you've heard mad cow disease, it's like that. So they are just proteins that are not folded correctly. You get a normal protein that maybe it makes your eyes green or something, I don't know. And it misfolds. Well, it no longer makes your eyes green because part of the shape of that protein imparts its function to it, which we learn in the chemistry chapter. So it's very important in biology for proteins to have the shape that they're supposed to have, not just the chemical makeup, but the actual shape of how they interact. The folds in this molecule interacts with that molecule, and then this one folds over and interacts this way, and they create this shape that creates the functional form of the protein. Well, if it's not doing that correctly, if it didn't fold up correctly, then we would call it misfolded. Yeah. And sometimes those misfolded proteins will stick to one another because they aren't behaving correctly. We talk about amphipathic, we talk about hydrophobic, we talk about hydrophilic. Hydrophilic, water loving. That's most of the things that are going to be on the outside of like cells, water-based stuff, or inside of the cytoplasm of cells is water-based. Anything that's hydrophobic, water fearing, so to speak, it's going to fold in on itself away from the water, right? And so a lot of times proteins fold in a way to have hydrophilic, the loving on the outside, and the phobic hidden away on the inside. Well, if these guys fold wrong and they've got the phobic on the outside, they're not interacting with the environment correctly. They'll stick together because they're trying to hide from the water because they're not folded right. And because they stick together, they create these plaques. And you can call them all sorts of things, but it's similar to amyloid plaques like you see with Alzheimer's disease. And when these plaques build up, it causes damage in the brain, including forming holes in the brain. So it's why we call it spongiform. So it's like a sponge, spongiform encephalopathies. So you may have heard of that term before. But that's what mad cow disease is. It's a spongiform encephalopathy. And it's one thing when it's in cows and it's BSE, bovine spongiform encephalopathy. But it's another thing whenever it's in humans. And we have Creutzfeldt-Jakob disease or variant Creutzfeldt-Jakob, which is like what we see whenever you eat meat that has mad cow disease and it gets transferred to you that way. It can occur naturally in people, and there's other diseases like that. But it's a prion. It's only misfolded protein. It's not got any genome. It doesn't have DNA. It doesn't have RNA. It doesn't have anything else. It's just a misfolded protein. So how does it propagate? How does it spread its disease? How does it do that? Well, it literally takes this misfolded protein with that nasty hydrophobic water fearing on the outside. It goes near your guys that, do its normal function, like we had said for an example, the green eye protein, right? It goes near those guys doing their normal job and causes them to fold up the nasty way. We don't know why or how that happens. Science doesn't understand that quite yet, but that seems to be what happens. And so that spreads that way. It's very hard to control something that doesn't have a genome. How do you sterilize something like that? So if you are dealing with samples that come from somebody that you suspect could have one of those diseases, a prion disease, a spongiform encephalopathy, right? Then you are going to treat it differently. You're not going to sterilize it the same way that you would for viruses and bacteria. You actually go like all out. It's going to be next level treatment. It's not important that you understand how it's done right now, but I used to work in Southwest Medical, and we had a guy that they suspected had this, and he had to have several samples over courses of days drawn, and it was this whole procedure, I tell you. But sterilizing is not as simple as just killing viruses and bacteria. Sometimes there are other factors to consider that are more difficult to get rid of. That's my point. So it just depends on kind of what you're trying to get rid of. All right. Incineration is the form of sterilization that we use to sterilize our equipment in the lab. We're literally going to take our inoculation loop, which is down at that bottom there. It's a little like wire on a stick, and it is a little loop at the end. And that's what you like touch it to your little bacteria so that you don't have to touch it with your hands. And it picks up that little sample for you. But you can't just keep going and using it from sample to sample, right? You have to make sure that it's sterile in between so you're not contaminating. That's the idea with aseptic technique. So in between everything, we are flaming that loop to sterilize it. Yeah, we're literally just burning it to ash. That is what we're doing, but that is sterilizing it. We'll even, like, as we're opening our tubes, we'll have our sample, our tube with our sample in it, and we will take off the lid with one hand and like put the tube into the fire for 2 or 3 swipes, and then use our loop, and then stick it down into the tube, get your sample, take it out, fire, 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 before and after, lid back on to keep everything as sterile as possible. Your sample is on here, put it on, I don't know, your plate, and then, fire it again. We're gonna have videos on this, and then whenever we get in the lab, don't worry, I will be walking through all of this. The concept, it comes kind of naturally once you start doing it and thinking about where the bacteria could be at any minute. But that's kind of what we're doing. We're following where the bacteria could be at any minute and controlling that for our benefit. We talked about media and what it is, just nutrients to help us cultivate our bacteria in our cultures. Agar, you might hear me talking about agar quite a lot. They'll be like, oh, on your agar plate, oh, put it on the agar. Agar is just something that we add to the media to make it solid. And it comes from algae naturally. So it's not solid like, like the table, but it's solid kind of like, it's not going to mush in like lip gloss. I'm just trying to think what a good example would be, but it's got a bounciness to it, kind of like Jello, but it doesn't give quite as much as Jello does. It's a little more solid than that. It's the best way I can describe it. Sort of like a gummy, if you eat like a gummy bear, like you eat one of those, like you can squish it and it doesn't break, right? All right. In order to study bacteria, you need to avoid contaminating. We've said this, I think, already 50 times, but hell, that's just going to mean that we're learning it more, right? Yeah, so aseptic technique, preventing contamination. We're going to keep everything around us and our tube itself. It says as sterile as possible, but preventing contamination is a better way of looking at it, right? Because is your tube with your bacteria in it sterile? No. And is your environment around you going to be actually sterile? So we say as sterile as possible, right? You're going to be working without gloves on and using your hands. Like, your hands are not sterile, and they're not going to be sterile even if you washed them, unless you're like, even if you're doing a surgical scrub, it's probably not sterile. It's very close to it, but that's why they still don gloves and everything like that. So we want to get it as clean as possible. But it will not be necessarily maintaining true sterility. Just keeping things from being contaminated is a better way of wording it. So we'll use Bunsen burners to keep everything that we're using controlled, sterile, the openings of the tubes and the loop that we're using to transfer things. And we'll clean surfaces like of our desk, or like if you spilled on the outside of your tube and it's dripping in your tube, we'll clean any surfaces that come in contact with the culture with disinfectant that we will have in lab. Don't worry about what it is. We'll talk about that in a different chapter. Can't you wait to learn about how disinfectants work? The things you didn't want to know. So now, yeah, and then washing your hands afterwards, just to be sure that you're not taking it home with you, right? These are all part of the aseptic technique. So here is basic, technique that I've already kind of walked you through just then with my beautiful demonstration with my pen and my phone. But clean your bench tops, come in, you'll squirt it. Literally is like a squirt ball, the ones that have like the little spout on it, and you just squirt it all over your bench top, wipe it off with a paper towel. That just goes in regular trash. Bunsen burner flame. If you're not good with using the Bunsen burner, no worries. We'll walk you through it. But we use those strikers to start the flames. And we will sterilize our loops using the flame of the Bunsen burner. All right, when opening your tubes, you do not ever, folks, do not ever, I better not see you ever setting your tubes down on the table. Because if this is, let's say this is your, culture of your Staph aureus, and let's say like in, the real world, in your hospital lab where they're actually doing testing like this, you have a tube that is a sample of resistant, or MRSA specifically. Let's even go with MRSA, right? A resistant form of staph. About 99% of all strains in the entire world ever, anywhere that you could look, about 99% of them of Staph aureus is resistant to some sort of antibiotic. It's penicillin first, and then after that, other things. That should be concerning. But anyways, moving on. MRSA, right? You have a tube of it, and you want to see what it's susceptible to. We'll talk about how that would work later, but when you take your lid off and you're holding your loop that you are sterilizing in your Bunsen burner flame, and now you want to put it in your tube, what are you doing with the lid? Well, I open it with my pinky and my ring fingers. You hold it with your pinky and ring fingers and then lift it off of your tube, just like I am the lid to this Sharpie. So just like that. And yes, it is important that you do it that way, because if you put your lid on the table and you have MRSA on this, and you're just like one of these people who's like, oh, I don't know what I'm doing, and you put it there, now you've got MRSA all over your table. Now you might be like, well, I'm going to disinfect it later. Are you going to disinfect it before you put your elbow in it though? I'm just saying, or before your, lab mate does, just like, let's just not do it. Or the other problem is you set your carefully isolated culture of MRSA to diagnose your sample, and set it on the table. The lid gets contaminated with something on the table. You put it back on, and now your culture is contaminated. Just don't do it. Okay. Never does the lid touch anything but the inside should never touch anything but the tube. Okay. All right. So you've sterilized your loop. You've taken off your lid with your pinky, and I promise it gets easier to do it this way. It seems awkward at first, but it ends up being, you'll get better at it. And then you'll flame the opening of the tube. That sterilizes that area and the air around it. In a sense, it's really silly, but it is kind of how it works. If there were any microbes kind of in the air. Are you breathing when you do this? Yeah, because you're breathing because you're a human being and you need to breathe in order to stay alive. But you're gonna go ahead and sterilize everything you can about this. So you're gonna flame that opening of the tube a couple of times, right across the Bunsen burner, just 2 or 3 times slowly. And then you don't want it super hot. The loop, you want it like bright orange. It's a piece of metal. You want it bright orange, like it's as hot as it can get without melting from the sun, right? So that'll be sterile that way. But the tube, it's glass. You're going to pass it through the flame 3 times. And now that we're sterile, you'll put your tube down into the actual liquid, pull it out. If you have the loop, because it's actual loop, it's like a circle, it'll look like you're going to blow a bubble with it. It'll like hold on some of that liquid there. That's all you need. I want to say probably the biggest, I don't say problem, but it's probably the most frustrating thing for me as a professor is that I will tell students whom I love, that maybe you don't need to grab the entire sample in something. Maybe you don't need the whole thing. And people just don't believe me. They just don't think it's enough. But yeah, no, it really is. If you see like one of the tiniest little colonies, the size of maybe the little period on your keyboard, yeah, you could take that loop, literally just touch that, and it would be enough for everything you're trying to do. I promise. So I will give people sometimes slants, which are just auger at an angle. They'll be in tube, like a test tube, and they'll be at an angle, solid surface. And it's just a way that we can control contamination because the plates are just big. You open them up and there's a lot of air coming in. So I'll give you slants to be working with in this semester. And they'll just scrape off everything off of the surface. And that one tube that I'll be giving you once we get started is the only tube you get the entire semester. So if you scrape it off at the get-go, you don't have any for the rest of the semester. And I warn people of that, and they just don't listen, and then they ask for more. And I'm like, that's literally all I have. Like I was telling you. So we'll get into that, and I'll remind you about it when we get started. And people always mess it up. I literally don't have any other of it usually, but the good thing about bacteria is that even when you can't see it, it's still there typically. Which is part of why I keep trying to tell people is like, okay, now you get to actually learn that even though you don't see that you picked something up on your loop, that it probably is there, because all of their, experiments continue to work because now they're touching this slant that looks like it's got nothing on it and still getting sample. And I'm like, do you see what I mean now? You can't see it on your loop. It's okay. So that's the point here that I'm trying to make is like, you might not be able to see it on your loop, but it is there. So just have faith. Okay. So we've got it out. We've we got our sample out on our loop. We don't need our tube anymore. So we'll flame it same way 2 or 3 times and then pop that lid back on and set our tube back in the rack so we can have our free hand, right? So we can open up our plate, take the lid off of our little Petri plate, and those are hard to work with without setting down. So you can either hold on to the lid and work with your plate on a table, or which is what a lot of people do, or you can set the lid down in this case, and then pick the plate up and do it like that. It just kind of depends. But those are usually, since they're temporary use, I'm not as concerned about that being put down on the table as long as they are flipped up when they're set on the table, you know what I mean? And then replaced immediately once you're done. We don't flame the plates, okay? It's just open, put the stuff on it, and then put it down. How do you put the stuff on it? A lot of people have an issue with that. The loop, I don't have one in here because literally they just took everything and I couldn't grab anything. I might have one in here. Let's see. I think I took all of them back to the lab. Make one real quick though, just for demonstration purposes so that you guys get this, because this is one of those things that's I don't know, not confusing all the time, but it can be. So I'm using a paper clip just for demonstration purposes, right? So the loop, not the best of my work, but imagine that this paper clip this is the loop. And sometimes it has a little bit hanging off on it. Sometimes it doesn't. It just depends on what loop you got. And then it's long metal, just like the paper clip. And then there'll be like, a stick that it's on or something. Yeah, that's what you hold. And this whole entire wire, when you're sterilizing, the whole thing, the whole wire part of it, the whole metal part, you need to get bright orange before every time that you flame it. Okay. Your sample, you will like literally have like a little colony sticking up on a plate, and you'll come in with your loop and just touch it and grab just a little bit of that colony. If you've marred the colony and you can see the colony isn't there, or it's been made gooey or whatever it is by your loop, you have it. You have enough. I don't care if you don't see it. Okay. Then you'll take this, of course, using your aseptic technique, and you will not, because you took your sample, typically you're going to take it like on an edge, right? So you're going to take the edge that you took it on and flat with your loop, just gently touch it to the surface and gently run it on the surface. Okay. You don't want to push. It's like a gummy bear. It will just eventually, if you push hard enough, cut into the gummy bear, you know? So it's pretty sensitive to that. You want to just lightly touch it on there and just lightly drag it across. And you see how I'm doing it in like a zigzag motion. That's pretty effective. Most of the time I'm going to have you put in a zigzag and I just draw out straight. You see, I'm not going over myself at all. But some of the stuff, it doesn't matter if you do go over yourself. It just depends on what we're doing. And as we get more familiar with this, you'll get an idea for that. But yeah, so once you're done, if I was done with this, you don't just put it away, right? You need to sterilize it first. You touched it onto your MRSA or whatever. You will sterilize it again. So hot, glowing red fire all the way. And then you will let it cool a little bit and put it back in your drawer or whatever. But yeah, once it's cooled, it's sterile. So you can put it wherever. Please don't just put it in your mouth anyway, just because of the thought of it. So this is a little video that we're going to watch. Yes, we're actually going to sit here and watch it together. I'm probably going to put it on a faster speed. I'm going to put on, we're going to try 1.5 if I'm feeling it. Otherwise, I'm going to bump it up to like 1.75. But we're going to see how this goes. This way you can actually see this happening since I can't do this right now. I don't have anything. Just FYI. All righty. Hopefully.
In this video, you will learn the proper practices and procedures for performing aseptic technique. Using aseptic technique, you will be able to transfer a bacterial monoculture into a fresh medium.
Yeah, see, it's 6 minutes. I don't want to sit through the whole 6 minutes.
In this video, you will learn the proper practices and procedures for performing aseptic technique. Using aseptic technique, you will be able to transfer a bacterial monoculture into a fresh medium.
I'm sorry, I restarted an accident. It's my fault.
Two of the most commonly used tools for transferring bacteria are the inoculation loop and the inoculation needle. Knowing how to use a Bunsen burner is an important skill in aseptic technique. To light a Bunsen burner, start by closing the valve at the base by rotating the collar so that the vents are covered. Then slowly turn on the gas. Once you hear the gas, ignite the burner and set the height of the flame by adjusting the amount of gas.
You won't have to mess with the Bunsen burners anywhere near this much.
Adjusted by rotating the collar to control the flow of oxygen.
But you will use the strikers.
The coolest flame is yellow and orange. A medium flame is blue. And the hottest flame is a roaring blue flame characterized by a clear blue cone in the middle. The tip of the cone is the hottest part of the flame. Inoculation tools are sterilized by passing them through the hottest part of the flame. Once sterilized, inoculation tools should not be set down on any surface. In this section, you will learn how to transfer bacteria from a liquid culture to a petri dish. After flaming a loop, take the lid off the container of liquid culture and pass them out through the flame. Then dip the loop into the liquid culture and flame them out again before replacing the lid. Transfer the bacteria to your petri plate. Lift the lid of the plate and hold it over the base as a shield to prevent contaminants.
So this guy's way rougher than I would ever be with this auger.
Rotating the plate a quarter turn between spreads will ensure that the bacteria is.
You don't have to worry about this part where he's like turning it and then doing it and turning it and then doing it. We'll talk about how to do each of your plates, but yeah, you will just put it on the auger. It's not, you don't have to be this violent either. Maybe it just looks that way because it's this faster speed.
In this section, you will learn how to transfer bacteria from a liquid culture to a liquid. After sterilizing the inoculation loop, take the lid off the container of liquid culture and pass them out through the flame. Then dip the loop into the liquid culture and flame them out again before replacing the lid. Now take the tube of liquid media and pass them out through the flame. Then dip the liquid bacteria into the tube. Flame the mouth of the tube before replacing the lid.
Oh, sorry, I didn't realize that clicking on that would make that happen. Just play it.
Flame the loop to sterilize. In this section, you will learn how to transfer bacteria from a petri plate to a stab culture. Start by sterilizing an inoculation needle by passing it through a flame. Allow the needle to cool. You can test for coolness by tapping the needle in a clear area on your auger plate. While holding the lid as a shield, gently rub the inoculation needle over a single colony. Transfer the bacteria to a culture tube filled with nutrient agar. Remove the top of the tube and insert the needle, making 3 deep stabs.
So
fish,
that's a
replace the lid on the
solid,
tube and sterilize the needle.
solid media in a tube like that. It's called a butt. Stab
In
in a
this
butt.
section, you will learn how to transfer bacteria from a petri plate to
Yeah,
a
I'm a
liquid.
kid. I'm a child.
Sterilize an inoculation loop by passing it through a flame. Let the loop cool. You can test for coolness by tapping a loop in a clear area on your outer plate. While holding the lid as a shield, gently rub the inoculation loop over a single colony. Now take the tube of liquid media, remove the lid, and pass the mouth through the flame. Then dip the loop with bacteria into the tube. Pass the mouth of the container through the flame before placing the lid.
So I want to say something, which is simply that if you watch how much of that colony he picked up, he just touched on it, right? You didn't even see it on his loop. And then when he went to put it into that tube, he just swished it like a couple times. He didn't get violent with it, anything like that. That's all it takes. That's all it takes for those bacteria, because when you touch that colony, even though you don't see it, I promise you there's millions of bacteria on that. That's how tiny they are. That's how many there are. And if you start seeing that bacteria is present, like in an infection or something like that, like on an open wound or whatever, that's a lot of bacteria. It takes a lot. So yeah, so think about it that way. They are there. They are super, super, super tiny.
Sterilize the inoculation loop to complete the process.
Like when you think about it, they talk about like those Demodex mites that are on your eyelashes, like everybody has, but most people have them. You don't see them, but there's little mites on your, God, I don't want to even think about it, but it is a thing. And your eyelash, like pores or whatever, and you can't even see those. And we're talking about bacteria that are way smaller than that. Just trust that they're there. They're there. All right. So this brings us to this concept of the biosafety levels. So we're trying to keep everything controlled, prevent contamination of our sample that we're working with, whether it's MRSA or whether it's just stuff that we're working with in the lab. Like we will be working with Staph aureus, but it won't be MRSA. It'll just be lab friendly, non-pathogenic. It's not resistant to anything. So yeah, so no matter what we're working with, you want to, or how you're working with it, you want to maintain aseptic technique. But is it always enough? Do you always feel like you'd be safe? And if I was telling you, okay, we're going to be working in the lab. You don't need your gloves. Here's your little Bunsen burner. Here's, your sample of Staph aureus. It's non-pathogenic. You'd probably be fine. And it's everything, whatever. We'll do aseptic technique and everything's fine. But if I hand you a sample of the plague, are you going to feel good about that? So this is why biosafety levels exist. It's to help protect us basically from pathogens that we want to do research on and understand more about in a safe way, to keep us safe while we're doing work on these organisms. You guys know about how this gets whenever it gets to that really high level BSL-4, where they're wearing the suits and they're pressurized and everything like that. So we're going to talk about that a little bit as part of learning about how all these laboratory stuff works. So BSL rankings. So at the bottom, we have BSL-1, probably the closest to our lab. We call our lab BSL-2. It can be BSL-2, but we're mostly practicing at a BSL-1 level. We say 1+, okay? Microbes are not known to cause disease in immunocompetent people. Minimal potential for hazard to laboratory personnel and the environment, right? So skin bacteria like staph, yeast, things like this that you are like, yeah, if you're making beer or something like that, you are still doing, work like a laboratory person would be doing. So that's BSL-1. BSL-2, now we have microbes that can have moderate potential for hazard to personnel. So we're talking about herpes, rhinovirus, RSV, salmonella, pathogenic strains of these, but we can control them and it's not super serious as long as we're practicing these sets of rules, there shouldn't be a risk, right? BSL-3, we have to start introducing special rules to protect people from aerosols or something that could be created by working with these things. So it could be indigenous, or they could be indigenous being like around, you normally see them around, or it could be exotic. It just kind of depends, but they have serious or potentially lethal disease that they can cause. Tuberculosis is an example. They work with this in a BSL-3 environment. It is a pressurized room so that the room itself is negative pressure, which means it's less pressure, so that if there were a break in the pressure system, air rushes in instead of out. So the work that they're doing on tuberculosis in won't get out. So it'll stay there, right? And air is circulated there, but through a HEPA filter so that it is filtered out, you know, and all that. And they'll wear little booties that they'll change before they go in and out. And they might even wear like disposable gowns in and out and have all these procedures that they'll go through to protect themselves. So no big seriousness. You've seen stuff like that before. But this is also where they would work with the plague. So the plague is the Black Plague, bubonic plague. That's where we would be working with it as a BSL-3 environment. Now, BSL-4, here, these are all exotic, pretty much things that people don't ever see on a regular basis. Aerosol transmission of this can lead to deadly disease, almost, seriously fatal disease. So Ebola, when we're talking about 90% fatality rate, if you aerosolize that, whenever you're doing a like hot little, loop into something, and then it goes like that, that can release the pathogen into the air and expose you to it. So they don't want that. So you can do your regular laboratory work. So they put you in a suit. So the room is negative pressure, right? And so if there were a break in the room, again, secure room, air would rush in to the room. So there's no Ebola getting out. And if you are working in there, you're wearing a suit and it's pressurized, positive pressure, so that if your suit rips, air goes out and the pathogen doesn't get in. So they use pressurization of air to protect people there. It's all sealed. Yes, they do tape, like the glove to the suit and everything like that. And it's all contained, and they go through chemical showers going in and out of it. The work that I was doing with the Army and the CDC, they were doing my actual live BSL-4 work for me, because there's very few labs that will do that research on BSL-4 pathogens. And so you usually have to find somebody that'll work with you. And I was lucky enough to get the big guys to work with me. So they were doing my work for me. And so when they were building their new laboratory, it's not new now, but this was back in like 2007. It was new then. The CDC was building their new laboratory. We went to go see that in Atlanta, and it was their BSL-4 laboratory, and they had like a BSL-3 suite like around the whole thing, and you had to have a retina scan to get into that, and then there was like a whole other procedure to get into the BSL-4, but they had 4 suites for the BSL-4, so 4 labs, basically four full labs, and they're walled off from one another, and there's chemical showers in between each of them, and in between getting into the BSL-3 areas. And they had that all for one of the suites. It was clean and it had been turned off, and they were just cycling the chemical showers for the rooms because they have to be able to sterilize those rooms individually, and they cycle in between in order to keep it as clean as possible so they can sterilize it every once in a while, just the whole room, just knock it out so you're not just getting nasty everywhere. You can't really clean it, so so that's how they did that. And they had just cleaned this particular suite. And so we got to go in it through the chemical shower, and it's like a pressurization chamber as well. And so you like feel the pressure, and we didn't go through the shower itself. They had that part turned off, but it's only one person really is supposed to fit in there. And we had like 3 at a time. It was so claustrophobic, but I can't imagine working in those suits. I thought that's what I wanted to do. But now, like, as I'm old now, and so I look back on that and I'm like, yeah, there's no way you could get me in one of those suits and like have me holding on to like one of those tiny little microfuge tubes, the ones that are like really, really tiny tubes, and working with those gloves and like the tiny micro pipette. No, not with that suit. No, and that air rushing the whole time. Forget it. I'm not doing that. So I guess it's better I got out of that before I got caught into that. But yeah, so that was not it was cool to go see it, but I don't want to do it. So if you guys want to do it, more power to you. It was not for me. It was cool to see it. So this is most of the time our lab is being used as a BSL-1 lab. We have controlled access. We have one door, and it does notify people that they're going into the laboratory before they come in. We have biohazard signs up, letting people know that there is biohazard risk. We have a sink for hand washing so that you can actually wash your hands and be clean before you leave and not carry what you were working on with you. We have a sharps hazard that's properly labeled. We already talked about dealing with that. Your PPE, personal protective equipment, that's just going to be your lab coat and making sure you have closed-toed shoes for our lab. We don't need goggles. We don't need gloves. If you want to wear gloves, you are welcome. Yeah, and if you're ever not sure what to do with your gloves, whether it goes regular trash or bio trash, you can always ask. But yeah, if you do, wear gloves. But it's not required. And then we have the lab bench that we'll be doing our actual work on, that will be disinfecting before and after. And then on the far right, that's what an autoclave that's built into a wall looks like. That's kind of like what I used at my last job. And the ones we have here are smaller. Excuse me, but that's how we're going to be sterilizing all of our bio trash and stuff like that. The sharps containers, the bio trash, and all of the racks of tubes that you guys don't need anymore are all going to go into the autoclave. It uses heat, pressure, and moisture, steam to sterilize, completely kill everything. And then we can just throw it in the regular trash after that. So the autoclave helps control that spread as well. So that's just BSL 1. In BSL 2, we're going to have basically the same thing, but there's just going to be a little bit more protection. And in here, they're showing as our protection. I don't know what is going on with my throat right now. Our protection here is number 4 in this. It's not paying attention. I'm sure you guys can tell I'm pretty scatterbrained. It's that's 3. All right. This guy here. This, like the safety cabinet or whatever. It's going to be a laminar flow hood. It just makes sure air flows in a way to keep the organism that you're working on from getting outside of that flow hood. We have one of these in our lab. We don't really use them. We would use them for pathogenic microbes. We have some pathogenic microbes in the lab, but we're not working with them with you guys. So don't worry about that. But if we used to, then we would use that to work on them. That's really the difference there. All right, so if there's more risk, maybe for protecting, you protect your eyes too. Maybe wear goggles in addition to what we do, but like whatever, need-based. So BSL 3, we do have risks here. So we have the same kind of setup, but now we have 2 doors that you'll go through, and there's a shower in between. So you can, sometimes you'll have a shower in between, so you can shower off afterwards. So you'll go in there, take off your little hood or your little gown or whatever it is, and shower off, and then put on your regular clothes, and then go out of that area. So that in between area, the staging area, where you can make sure that you're getting cleaned or whatever. So there's a personal shower out. There's also going to be a powered purifying respirator that you sometimes will need. They definitely use this in the Army. Army, man, I'm just telling you, they stretch some rules. But the organism that I worked on was Crimean Congo hemorrhagic fever virus, or CCHFV. Wow. Anyways, it was BSL 4 to everybody except for the Army. Army worked on BSL 3+, and they wore this mask, and they said that was enough. Whatever, man. Like, whatever you want to do. It's not my problem, right? So yeah, so they would use this respirator that they would wear while they were working with it. Some of these other guys, like if you're working with HIV, which is a BSL 3, just due to the dangerous nature of the virus. Yeah, basically, if you were working with that, like you don't need a respirator to protect you from HIV. So it just kind of depends on what you're working with. Another thing you could have, you're almost always going to have a HEPA filter to the whole room to keep the air clean, and then an effluent decontamination system. Anything that you're vacuuming out, like from a liquid or anything like that, you're not going to be moving those liquids in and out of that room. You'll be moving it to that special chamber where it will decontaminate anything. So it'll be filled with bleach or something like that. But it is a containment system in and of itself in like a separate room and all that. Yeah. So, yeah, and then it'll be usually air controlled, and you will disinfect it, and it won't be exposed to any other portion of the building that you're in and everything like that. So that's just BSL 3. I think it's about HIV if you don't understand about it, and we will learn about it in a later chapter. HIV is, I don't know, something. I don't know if I want to call it beautiful or, artwork, nature's artwork, or terrifying or horrible. It's all of these things, but it's a virus, and it has RNA as its genome. And then what it does is it turns its RNA into DNA using a special enzyme called reverse transcriptase, which basically flipped our understanding of biology on its head. And when we discovered this, and we've used it as a tool actually in biology now. But yeah, it went the reverse way of what we were used to, made DNA from RNA. And then it takes its little DNA that it made, yeah, and it puts it into your DNA forever. How about that? It's not the only one that does that, by the way, but it's the only one that goes in the reverse to make its copy and then do that. Not the only one, but the one that we'll be talking about. Other viruses do this, like herpes and stuff like that. They do stuff like this, but they don't need to reverse it first. So yeah, there's viruses that stick their DNA into your DNA. There's no getting rid of it after that. That's why we say that they're forever. Herpes is forever because it sticks its DNA into your DNA forever. How do you get that out? Into your DNA, like it's part of your DNA, your genes now. So that's what HIV does. So that's why you can't cure it. You can treat it and have no virus load. You can test somebody's blood and say this person isn't showing any presence of any virus. You stop their medication and they will have HIV again, 100. They didn't clear it. They're not done. It's still there. It's just in their genes. HIV, man. All right. BSL-4, the difference here when we are comparing to the BSL-3, we're just having enhancements of the BSL-3. So we might have more shower stations. We're going to actually, like our protective suit, we're going to actually shower the suit off. That's what I was talking about with those chemical rinses. You put the suit on first, then you go in the chemical shower, then you go into the lab, and then you do your work. And then when you're going out, you go through another chemical shower. And then take the suit off, and then you'll shower your body off, and then you can go home. And it's that sort of a thing. So there's just more in there. There's the pressurized suits that you're going to be working on. So that'll just maintain your safety there, and it'll always have airflow, and there'll be little like guys hanging out from the ceiling like this, so you can hook on to them wherever you're working at and maintain that positive air pressure. It's a one-way flow valve, so when you unhook, it's not like you're a danger to getting contamination. But yeah, that's basically how that's going to work. Yeah. All right, so that's the idea that's just relating back to our aseptic technique and all of that, so that we can understand a little bit about why it's important to prevent contamination on a bigger scale. So whatever, isolation. Most of the time our samples are mixed, especially when you're collecting it from a patient, like our wounds that we were talking about. So if you're only interested in one of the bacteria that you're collecting, the one that's causing the disease, how are you going to get it separated out so that you can work on just that bacteria and identify it and learn how to treat it? One of the most useful things, and probably the one that you need to know for the lab, and I always ask a question about it on both of the tests for the lab, okay, is the quad streak plate. It's pretty easy way to spread out bacteria, sort of like diluting it on the surface of a solid plate. So we're not diluting it with liquid, we're just diluting it by spreading it out. So imagine that you have a marker that's running out, and you first put it down on your piece of paper, and as you stretch it out, and it gets fainter and fainter and fainter and fainter, right? That's what we're doing with the bacteria. We're getting a little bit of bacteria and spreading it out. And as we do that, it's getting fainter and fainter and fainter and fainter. Then we'll sterilize our loop, turn our little plate, and from the fainter area from before, with our sterile needle, we'll put it in there once, maybe twice, and then fainter, 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 fainter again. So we're diluting it every time that we do that. So the whole purpose is to dilute it out so we can separate out the cells more and more and more and more and more till eventually we have individual cells separated out. And what do individual cells grow into? Colonies. And we can pick one of those and create a pure culture from it. So that's the idea. So here's how it works. Step one, you're basically just putting your sample down. Honestly, in step one, it doesn't really matter how you do it. You just are going to take your sample and you're just going to somehow. It doesn't even have to be perfectly zigzagged out to dilute it. You can even go back and forth if you need to. You're just putting it down. Okay. Just putting it on the plate. That's all you need to worry about for step one. Step two, Now we're going to start worrying about diluting it out. You go into the first quadrant 1, maybe 2 times, and then spread it out with a few zigzags. You're flaming your loop before you start doing that so that it's clean, right? Then you're going to flame your loop again after you do number 2. So it's clean. So when you go to do number 3, you're actually just diluting it out. You're not just adding to what's already on your loop, right? So we're diluting it out. In step 4, this is our final dilution, if you will. You're going to get that sterile loop again, get it into that quadrant 3, the last quadrant that we did, again, in that diluted area, and then zigzag out all until that whole rest of the open space of that plate, take up the whole rest of the plate. And that is where you're going to be more likely to see your single colonies because it's the most spread out there. So we do have a video. This one's pretty short, I believe. Just so you guys can see it again, because this is one that, like I said, it's always on the test. I don't want laser focus. Geez, that's big. Can I just not have? There we go.
Pure cultures of bacteria are most often obtained by the streak plate technique. In this procedure, an inoculum, a mixture of 2 bacteria in nutrient broth, is placed on a solid nutrient medium and then repeatedly diluted to separate individual cells. You will inoculate the agar using a 4-quadrant streak as shown. Use a fine-tipped Sharpie to divide the bottom of the nutrient agar plate into 4 quadrants. Label the bottom of the plate with your initials, the date, the type of medium, and the culture or source of the inoculum. You don't want to label the top of the plate because tops can get separated. Use a sterile loop to obtain the inoculum. We will use a loop full of Staphylococcus aureus. Flame your loop until it is red hot and let it cool. Remove the cap from the tube, pass the tube through the flame, obtain a loop of bacteria from the tube, and then pass the tube back through the flame before recapping. Lift one side of the Petri plate lid and streak about 1/4 of the agar with as many
Does it better than I do. I
streaks as
can't lift the
possible.
lid like
Hold the
that.
loop like a pencil and gently touch the surface of the agar. Replace the lid and flame the loop. In the next steps, we will spread the bacteria from the initial inoculum into subsequent areas of the agar plate. Always let the loop cool before performing the streak. Using the sterilized loop, streak through one area of the first sector and then streak a few times away from the first sector. As shown in the illustration, there is only one streak through the initial inoculum. Replace the lid and flame the loop. Using your sterilized loop, streak through one area of the second sector and then streak a few times away from the second sector, as shown in the illustration. Replace the lid and flame the loop. Using your sterilized loop, streak through one area of the third sector and then streak a few times away from the third sector, as shown in the illustration. Replace the lid and flame the loop. Incubate the plate in an inverted position at 35 to 37 degrees Celsius for at least 24 hours. During incubation, water evaporates from the agar and will condense when it reaches the cooler Petri plate lid. If the plate is inverted, this condensation will stay in the lid, away from the agar. If the plate is incubated with the lid on top, the condensation will rain back onto the agar. Bacteria will move in the water, and you won't see discrete colonies. Individual or small groups of cells will divide, producing a visible colony-forming unit, or colony. Observe areas of well-isolated colonies. Different species will produce colonies that appear differently. This plate has a pure culture. All the colonies are identical. There are 2 different bacteria on this plate. One produces red colonies, and the other makes smaller yellow colonies. After recording all of your data, properly discard your plates to be autoclaved.
Okay, so you can see how, especially in that second plate with the different colored colonies, that could help you separate out your samples just from different, from one another. But that's the idea of that. We don't need to see that again. Okay. Yeah, we could review. So I'm supposed to review the pre-lab content with on Top Hat. That was because the idea was that you guys weren't going to be able to get on Top Hat. So now that I know that you can, we don't need to review that together, really. But yeah. So so that part's fine. You could watch this next video. This one's kind of a longer one, but it's basically he's doing a urine culture. So he is using these principles to do testing on a urine culture to determine, you know, what the bacteria is for a patient. It's easy to tell for me whenever I watch it. Like I already know, like from like the first like minute in, like what bacteria it was going to be based on it, how it grew. But sometimes some of the media that we use will add something to it to prevent some things from growing. and therefore other things can grow that we're targeting. And if they can grow on that, it's a sign that it is XYZ species. And if they can't, then it wasn't XYZ. And then you can use that information to help you determine what the organism is, or what kind of color changes you'll see for the media or anything like that. So using different kinds of media with different additives and different nutrients and whatever, and how they grow on it and cause color changes can help you determine what organism you're dealing with. And it can be better to do it that way than to waste your time sorting through different PCR protocols that you might have. So there's that. So I'm not going to watch this one right now. I am going to post this PowerPoint probably so that you guys can access this if you wanted to. But it's like 12 minutes long. So, all right. So aseptic practices don't just play a part in culturing microbes. Anytime you're scrubbing for surgery and creating the sterile field in surgery, that's creating aseptic technique, but in the surgical, suite. Then washing your hands after using a restroom or before you make dinner, wearing gloves when you're touching patients, and making sure that you change your gloves whenever you're switching between patients, and not sharing needles between patients or using the same medical supplies over and over again. All of this is just aseptic technique. It's just in the medical field, right? So just think about it as being similar to that. All right, so the worksheets that we typically use in the lab that you guys would be submitting as your post-lab thing. You can access these on Top Hat, or you can access them on Canvas. On Canvas, it's all of the worksheets all together. We went over that being in the lab worksheets and documents area in the modules area, right? It's all in one, it's like a lab worksheets or something. On Top Hat, each worksheet is for each exercise. So on the front of the worksheet, the first page of the worksheet is all the information basically that we went through just now, as well as the procedure and steps. And it's what you would have like in our little dry erase insert thing. So the front page would be all the info, and then the back is this worksheet. And this worksheet is what you turn in for credit for that. So this is our first one. So I'm going to go through it with you. There's usually 3 questions and then a grand rounds exercise. Okay. So here we have explain how our lab practice of disinfecting our bench tops, both before and after the lab period, relates to the importance of aseptic technique. So we're trying to prevent contamination. That's what you should always think when you hear aseptic technique. How is that helping with that? Removing any possible source of contamination that could get into our stuff that we're about to work on, and then removing any product of our thing that we were working on from being contaminated later on for other people, right? So that's how that will work. Why do we flame the loop between the quadrants of the streak plate? Why don't we just keep going with the same loop? Because we're not trying to keep spreading it out the same concentration. We want to dilute it out. So every time we're flaming it, sterilizing it, setting it back to 0, completely clean. Now when we drag back into the already diluted area, we're diluting it even more because we're trying to get individual cells. That's the goal. And then in which quadrant of the streak plate would you expect to see the most individual colonies? The 4th. When you guys are answering these questions on these worksheets, I don't need full sentences. I don't need full explanations. Just get the idea across with a few words and you're fine, okay? And then we have a little bit of story about why this is relevant. We kind of already talked about stuff like this. And then the grand rounds. Typically, you'll go around to the different tables and go see what everybody's results are. Obviously, this first one, you wouldn't have results, but that's typically what the grand rounds are about. So you can see the different possible results. Your table's going to get one result, and then they might get a different result. And then the test might have the result that you didn't have at your table. So you better go look at the other samples because that's your moment to go see what the positive might look like instead of the negative. You know what I mean? So that's what the grand rounds are for. All right. So you guys, if you want to, I don't care for this particular worksheet. If you want to just take a picture of this slide, and I am going to be posting it online as well, and then post that as your evidence for your post lab for this lab exercise, I don't mind because it's not like you're going to get a different answer, right? So I'm not really that worried about that. Just saying that you were here and you sat through my BS and whatever. So if you want to take your screenshots now, for your evidence, this is probably the best place to do that. Yeah. So I'll just give it just a minute for you guys. And I am going to post these so that you guys can access them if you need to. Okay. Everybody good for me to go? All right. Okay, so that's it for today. There is online stuff to help you become more familiar with this that I highly, highly, highly recommend. There are assignments that say practice lab simulations not for credit at like, I don't know, it's like at the bottom of the assignments if you sort by type, right? They're not on any due dates. They are not worth credit. These are the ones that go along with this exercise. Aseptic technique, broth to plate, aseptic technique, broth to broth, and then isolation quad streak. Those are the ones that you're going to look for if you want to get practice in and interact with it. Since we're not doing these in the actual physical lab, I highly recommend that you knock these out. They don't take hardly any time at all, and it's not worth a grade. So you might as well knock it out at least sometime before the lab exam, right? So be aware of that. Those are always going to be there in the bottom of the assignments. So yeah, but that's it for today. I know I said it was going to be short and I guess I lied. I thought it would be. But that's it for today. That's all we're doing. Be sure that you put this in Top Hat where it says in post lab. That's what it says. Yeah, post lab. Just upload image there. And then that just shows on Top Hat that you did it. And then I can mark it off as complete and then put the grade into Canvas. So, yeah, so be sure you upload it onto Top Hat. Yes. Okay, cool. All right, any questions?
Yes, ma'am. I have one question.
Yeah.
With when you said that, like the HIV and the herpes, it's like DNA and DNA. You had mentioned the viral load, and I like with Hep C, there's a viral load as well.
Mhm.
So is that like in the DNA as well with hepatitis C?
No, so hepatitis C is not in your DNA. A viral load is just a term that is measuring the amount of virus in your body. So that can
Right.
be from any virus. So you could have, COVID and have a viral load. You could have
Okay.
rhinovirus and have a viral load.
Okay.
But yeah, so when they look at HIV, it's hard to tell, right? So you could be on medications for HIV and you won't test the virus being in your bloodstream.
Mhm.
But even though they don't see the virus in your bloodstream, you still have it hiding out in your DNA. They
Okay.
can test for antibodies in your bloodstream. And if you have antibodies against HIV, it means your body tried to attack HIV, which means you have HIV.
Okay.
So they can do that. So they usually do that to test for it instead of testing for the virus itself because it's not very accurate because it can hide, right?
Right.
So, but yeah, that was a good question.
Okay. And then one more question, I'll leave you alone, let you go. So on the questions that we did on the Top Hat, I know you said you'll go in and put the grades in and that's fine. But on the ones that where like, it has said like, what is a colony exactly? And I like put in my own words, it came as incorrect. Do you go back in and fix those or?
Yeah, I can go back and look at that.
Okay. And then the one about what is the purpose of sterilizing the loop between quadrants? I put it lowers risk of contamination, and that one came out wrong. So I guess that one is wrong then, or
I don't know. Like, it's just one of those things where it's like that part is touchy. So
okay.
I'll look back through it. No worries.
Okay. Thank you so much. I appreciate it.
Yeah, no problem.
All right. Bye-bye.
Thanks, guys.